Thank you very much for inviting me to be a presenter here, and thanks for the previous speaker's wonderful talk. So today, as Yuri mentioned, I'm currently a postdoc in Dr. Connor Evans' lab at Wellman Center for Modern Medicine at MGH and Harvard Medical School. Today, I will kind of show some of our work that has been done in our lab using coherent Raman imaging and have focus on the stimulated Raman imaging technique. So currently in our lab, we have multiple coherent Raman imaging system. We use different type of lasers, and those lasers has different type of performance. So we can build our system based on different applications needs. So it's a kind of customized system for the application that will be studied. One main application field in our lab that we use our coherent imaging system is to study the skin. Our lab not only interested in, so our lab is interested to use coherent imaging to study the skin composition, microstructure, and also we are interested to study after we apply the topical formulation on the skin. And then we will see how the drug permeates through the skin. We are interested in not only the pharmacokinetic, but also the pharmacodynamic of the topical drug. As mentioned in the previous speakers' talks, Raman imaging has the benefits of using the intrinsic molecular, chemical molecular information from the drug that we are, or the skin that we are, skin component that we are interested to measure. Because of that, we don't need to label the molecule, and we can image the skin non-invasively. And also using the confocal microscope setup, we can get a high cellular level resolution while we do the imaging. In addition to those advantages that we have for the general Raman imaging technique, the coherent Raman imaging we usually refers to CARS and the SRS. So for the coherent Raman imaging, we have the additional advantage of the fast imaging speed. So we can acquire the image very rapidly, and we can look at the skin and the drug permeation process in a video rate time resolution. In coherent imaging system, in coherent Raman imaging system, we need to synchronize lasers, and one laser called pump laser, and the other laser we call Stokes laser. And then we can tune those two lasers wavelengths to match the vibrational band that we are interested in. For example, if we tune the wavelength difference to match the 2250 wave number, then we can probe the nitrile group of the drug molecule, and then we can collect image from this drug molecule. In addition to image the drug or the skin using single wave number, we can also use SRS microscope to do the hyperspectral imaging, which refers to we can tune the laser wave number step by step, and then we can collect the spectrum. We can collect the image at different wave number, and then we can acquire the full spectrum information of the skin and the drug. Here are some image examples showing that we can use our the previous works that we've done using our coherent imaging system to look at the ex vivo mouse ear skin, and then show those, and we can visualize those different microstructures at different layers of the skin using the lipid wave number. And then we have also imaged ex vivo human skin, and then we can see those microstructures in human skin samples. In addition, we also use coherent Raman imaging to visualize the field melanin distribution in the skin. The left-hand side two images, the top one is the ex vivo mouse skin. And we can visualize some of the field melanin present in the sample. And the bottom one is the in vivo mouse skin sample. And we also took a human melanomas sample, and then we image using the coherent imaging system, and we actually observed some of the field melanin present in this sample. More study, we use coherent imaging system, so we also use the system to track or monitor the skin microstructure change during inflammation progression. So we use, in this experiment, we actually we use in vivo mouse model. And then we look at the stratum corneal layer microstructure change during the progression. So from top to bottom is from at the beginning of the inflammation or before, so the top row is before inflammation, and this is early stage, and the bottom one is the later stage. And we can visualize that while during the progression of the inflammation, the border of the cornea site become more irregular, and we also observe some cell swelling during the caused by the inflammation. So the previous slides, I showed that our lab have used coherent imaging to look at the skin microstructure and the different skin components. We've also, so this slide is a study that we use coherent imaging to look at topical formulations. So we are interested to see how the API permeates through the skin. And this study uses ex vivo mouse model. So for this study, we imaged ruxolitinib using the nitrile stretch. And then this video shows the drug permeates through the drug distribution change with time goes by after we apply the drug on top of the skin. And then in this study, we while we collect the signal from drug, we also collect signal from lipid channel, which helps to show the microstructure of the skin. And then we will use this microstructural information, and then we will extract the drug signal at the different microstructure region, so that we can get the intensity profile related to each of the region. And then we can compare the profile, the permeation profile difference between different regions. I think this study provides a view of using the coherent imaging system for studying the bioavailability of topical drug, and also because we can separate the signal from different microstructural regions. It would be helpful for study the drugs permeation pathway, so we can know better this formulation it prefers go this pathway, over the other one, etc. Yeah. In a more recent study, we used coherent imaging to look at the bioequivalence between topical products. I will use this study as example, and then I will go through details of the experiments and the results we got, and then so kind of show you some just the process that we will use, how we will use the coherent imaging for topical drug and skin study. Yeah. And then before I go to the experimental part, here is a disclaimer. This project is supported by the FDA of the U.S. Department of Health and Human Services as part of a financial assistance award totaling 1.25 million with 100% funded by FDA HHS. The contents are those of the authors and do not necessarily represent the official view of nor an endorsement by FDA HHS or the US government. Yeah, so this is the disclaimer. Okay, move forward to the slides. Development of a generic drug is important to help improve better access to the treatment by provide a more cost-effective option. When developing a new generic drug or looking at or confirming the current existing generic drug, it is important to assess or to evaluate whether the generic drug or the tested drug are bioequivalent to the existing brand name or the reference listed drug. Specifically for, according, as mentioned in the FDA recommended studies, for tazarotene topical cream, to evaluate the bioequivalence, it is important to show that the tested drug shows a equivalent total cumulative amount and maximum flux to the reference product. And we can use a pharmacokinetic approach. Specifically, we can use the maximum concentration Cmax value and the area under curve value to help assess the bioequivalence. The currently widely used method for bioavailability and the bioequivalence study is the IVPT study. Is the IVPT method. This method is well established and widely used, but it lacks the information of those microstructural information. Because of that, we cannot look at the spatial distribution of the drug within those microstructures, and also it's hard to look at the drug distribution at different skin layers. And then because the coherent Raman imaging technique, specifically the stimulated Raman scattering imaging technique, it has the good temporal and the spatial capability. So we can use this technique to fill the gap to help us to look at the drug, and at the same time we can show the spatial information. In this study, we particularly look at tazarotene, and we studied topical products that contain tazarotene as the active ingredients. Tazarotene has a strong vibrational band that locate around 1590, and then in this study we will use the single vibrational peak to measure the tazarotene signal. In this study, to better use coherent Raman imaging for bioequivalence study, we developed a reference polymer film. So the purpose of having this polymer film is we want to have a standard material that can help us to remove the day-to-day signal variance, and also can remove the signal of fluctuation caused by maybe the optical system, some interpretation, or maybe some signal fluctuation caused by the electronics that we were using. So and then we will so this polymer film can track this signal fluctuation, and then later when we measure the signal from topical drug applied to skin, we will normalize the topical drug's signal to the standard signal, so that in this normalization process we can remove this effect and only keep the signal that is related to the drug permeation. And then this is an image of the polymer film that we synthesized. And then we use PPMA as the polymer base, and we embedded tazarotene in this polymer film. In this plot, the red dot shows the background signal fluctuation caused by the reasons I mentioned. And then we can see that there are some signal variance during our long-term repetitive measurements. And then the blue dots as a signal comes from the polymer film. And we can see that polymer film can track the signal fluctuation, and it kind of matches with the background signal fluctuation. This slide shows gives an overview of the process we use in this bioequivalence study. In this experiment, we use ex vivo human skin. We will trim the skin to about a couple of hundred micrometers thick, and then we will separate into 3 segments. And the one segment of the skin will be applied with formulation 1, and a second segments will be applied with another formulation. And the third one we will put on the polymer film. And then those 3 skin samples will be positioned in 3 wells, and we image in our inverted microscope setup. This is the SRS microscope system we have in our lab. And then we will image those 3 samples continuously for a total of 6 hours. And then the measured intensity profile from the drug applied skin will be normalized to the intensity profile from the polymer film. And then we can get those normalized intensity profile for each formulation. Using the profile, we can calculate the Cmax value and AUC value. For each formulation, we will repeat multiple skin donors, and for each skin donor, we will repeat multiple samples. After collect all the data and have all the calculated Cmax and AUC values, we will do the bioequivalence analysis by comparing the AUC values of the 2 formulations and the Cmax values of the 2 formulations. And if the ratio of the 2 formulations are within the bioequivalence limit, then these 2 products or formulations bioequivalent. If it's outside the range, then they are not bioequivalent. In this experiment, we have totally 5 treatment groups. And then for the first group is the reference listed drug group. And then the second, so we have another group that also used reference listed drug, and we labeled R2 to distinguish from R1. So this is mainly to provide a measure of the inter-experiment variability, because they use the same formulation, and it's mainly to kind of help to show the inter-experiment variability. And we have the generic product group and the 2 alternative formulation group. The alternative formulations I'm added to have a group that should not be bioequivalent to reference listed drug, because we use gel or PAC 200 excipient composed formulation. So those very different compositions should lead to different permeation behavior. And more experimental details, I will just briefly go through those. The concentration we use for the study is 0.1%, and we have 4 different skin donors. For each imaging, we will go several different depths from the surface to 64 micrometer deep. And then the whole study duration is about 6.5 hours. And the skin sample are kept in a stage top incubator, and we control temperature to be 32 degrees Celsius. And here are the SRS image we collected in this study. And we can see that, so those blue images are from the drug wave number. And we see the drug distribution change after we apply the drug on the skin with time goes by. And at the same time, we collect the image from the lipid channel, which helps to show the microstructure of the skin. And then and then we will use this microstructure and then extract the drug signal in the lipid-rich interlaminar region and the lipid-poor corneal side region. And then we will collect and then we can obtain this intensity time profile in those two different regions. The left-hand side is the lipid-rich region and the right-hand side is the lipid-poor region. And for all the 5 test groups, those are the intensity time profile. The reference listed drug, the generic drug, their intensity profile has very similar intensity. For the alternative formulation, the gel formulation, the intensity is slightly higher than the generic drug and the reference listed drug, indicating a slightly higher amount of drug present in this treatment group. And for the alternative formulation PEC 200, it shows a much lower intensity, indicating the lower amount present. And in this study, for the lipid-rich and the lipid-poor region, their intensity time profile are very similar. We don't see a very distinct difference between the two regions. And then use the profile, we can calculate the Cmax, the Tmax value, Cmax value, and AUC value. We can use those values to do a pairwise comparison. And then for Tmax value, we observed that the alternative formulation composed of PEC 200 shows statistically significantly different Tmax value compared to the generic and the reference listed drug. And for the Cmax and the AUC value, the PEC 200 group shows the statistically significantly different values compared to the rest of the groups. And this is the main part of the bioequivalence study. We also use the calculated Cmax and the AUC value to do the bioequivalence assessment. Because we use the SRST technique, which provides the spatial information, so we can collect the Cmax value or AUC value in those different microstructure region, and then we can do the bioequivalence analysis in those regions separately. Like we can look at them respectively. The first row we compare the reference listed drug group 1 and group 2. And then we do this, we calculate the 90% confidence interval for the mean difference of the Cmax value. And we can see the calculated range is within the bioequivalence limit, 0.8 to 1.25. And then this indicates the bioequivalence of these two groups, which is as expected. And also this results confirms that our method has a good inter-experiment variability. And the similar trend bioequivalence results are observed in all regions and also for the AUC values. We also compare the reference list drug to generic drug, and then the value are also within the bioequivalence range. Here we observe the PEC 200 alternative formulation. When we calculate the confidence interval, the range we got is outside of the range of the bioequivalence limit. So this means that the PEC 200 group is not bioequivalent to the reference listed drug. This pretty much conclude this study, and we kind of use this experiment and the results to show that we can use our SRS microscope to monitor permeation of APIs, and we can use the collected profile to calculate the Tmax, Cmax, and AUC value. And specifically, when we use Cmax and AUC value, we can do the bioequivalence assessment to calculate the confidence interval to decide to determine whether two topical products are bioequivalent or not. Hopefully, this example can show you some of new information or some of different view of using novel technique in study scheme of topical formulation. So in previous studies and also in the bioequivalence example, we mainly use a single wave number to image the skin or to measure the drug. This has a requirement is that the single vibrational mode or peak should have a very strong signal, much stronger than the excipients in the topical products or the skin background. However, it's not always available for a lot of different APIs. So moving forward, our lab focused on using sparse spectral sampling SRS S4S method, which will look at multiple spectral peaks or even spectral segments to image the drug instead of using only single one wave number. In this way, we are not limited to those drug that has very strong signal in one wave number. So we can really expand to image more drugs, and then we can study more topical drugs on skin in the field. Yeah, here are example showing that we can use SRS spectral to show to distinguish three APIs that has very similar structure, but we can differentiate them by different peaks. Yeah, that concludes my presentation, and I want to acknowledge the funding agency and also the very great and excellent collaborators from FDA. And this work, the bioequivalence work, is contributed. Some of the past lab alumni, Fortis, Isaac, and Xiaolei, their contribution to the bioequivalence study. And also I want to acknowledge Professor Evans supporting this study. With this, I'm happy to take any questions. Thank you.